Okay, welcome. Today we are going to do the onion mitosis lab. So let's get started. The first thing is we have to label 2 of those tiny little cups. One with HCl and the other one with carnauba solution. And then we actually put the respective solution in there, and you're using about 2 milliliters. And maybe when you pour HCl, you might want to wear some goggles. So we have HCl here. And this is just a tiny little bit, you don't really need much. That. And then the carnauba medium. Or solution, about the same amount. We are going to take an onion root tip, which is in this glass vial, and with forceps we're going to pick one out, and you're going to place it into the HCl. That is going to soften the root tip so that in the end we can smash it under the microscope slide. And you leave it in the HCl for 4 minutes. Okay, so then once that is done, you take the onion root tip and you place it in the carnauba solution, which is a fixative. And again, you leave it submerged in there for 4 minutes. Okay, so now we are ready to make our slide. So we take the microscope slide, and we have done that now a couple of times. I like to clean it at the beginning. Just use a little bit of ethanol and rub it clean. Now we are going to take the actual onion root out of the carnauba solution. And you need to identify that the actual tip, which is the rounded side. So you put it on here. And with a scalpel, be careful, you're going to cut just about 1 to 2 millimeters from the tip, and the rest you can discard. And now you have the onion root tip right there. Now that we have the onion root here, we are going to take toluidine blue to stain the different parts of the cells, and you just put 1 or 2 drops on top and incubate for 2 minutes.
2 very boring minutes later.
After the 2 minutes, you're taking some blotting paper, some Kim wipes, and you're carefully, so that you don't remove the actual onion tip, it's at this point really small, you're going to take it and you just blot some of that toluidine solution as much as you can. Now we have a little bit of water here, and as last time we're making a wet mount, so we take some water, a couple of drops, and put it right here on the onion root, like that. Now the fun part starts. We take the cover glass and we place it there, and you can go from the side here so that it starts to adhere the water, and then slowly let it drop. Now we need to squash the onion root. You take another one of those papers and you put it right here, and with your finger you just literally push down. Make sure that you don't twist around, okay? Just push down and you're done. Now it's ready to be seen under the microscope. So pretty.